Have you ever wondered what you're really made of? I mean, if you could zoom in way past your skin and your cells, what would you even find? Well, you'd find these incredible molecular Legos that build, well, everything. Today, we're diving into the three master materials that construct all of life as we know it.
So, let's ask it again. What are you really made of? It's a huge question, right? But the answer, believe it or not, starts with just a handful of molecular blueprints. In this explainer, we are going to unpack the three master materials that form the very foundation of you, me, and every living thing. I want you to imagine life as the most complex, most incredible Lego set you've ever seen. The fundamental bricks, the pieces you build everything with, are really just three types of molecules: carbohydrates, amino acids, and the proteins that they build. If you can get your head around these three, you've pretty much grasped the core of biology. Okay, now here is the one rule, the single most important idea you need to remember for everything we're about to cover. Ready? In biology, a molecule's shape is its destiny. Its specific structure, its form, determines its specific job. You change the shape, you change the function. That's the key to the whole thing. All right, let's kick things off with carbohydrates. These are by far the most abundant organic compounds in all of nature. You probably think of them as fuel, and you're right, but they also provide really critical structural support in the biological world. The clue is hiding right in the name, carbohydrate. It literally means watered carbon, because on a chemical level, that's pretty much what they are, carbon atoms attached to water molecules. We're talking about all the essential sugars and starches that power us. We actually classify them by size, and a Lego analogy works perfectly here. You start with just a single Lego brick. That's a monosaccharide, like glucose. You snap a few of those together, you get an oligosaccharide, like the sucrose in table sugar. And when you link hundreds or even thousands, you build a massive polysaccharide, like the starch you find in potatoes. Okay, so this leads to a really fascinating puzzle. How is it possible for two molecules to have the exact same chemical formula? I mean, the same number of carbons, hydrogens, and oxygens, and yet be totally different sugars? This slide right here is the perfect proof of our main theme. Just look at glucose and fructose. They have the exact same parts list: 6 carbons, 12 hydrogens, 6 oxygens. But a tiny, tiny tweak in how those atoms are arranged, one forms a 6-sided ring, the other a 5-sided one, completely changes everything. One becomes blood sugar, the other becomes fruit sugar. See, it all comes back to shape. So, if carbohydrates are life's fuel, our next building blocks, amino acids, are more like its alphabet. And get this, there are just 20 common letters in this alphabet. And with those 20 letters, our bodies can spell out the instructions for pretty much every single biological process. Every single one of these 20 amino acids shares a core structure. It has a basic amino group on one side and an acidic carboxyl group on the other. It's this two-faced acid and base nature that gives them their incredibly unique chemical powers. Think of them as the individual beads that we're about to string together to make proteins. Now, from a nutrition standpoint, they fall into two basic camps. The non-essential ones are the ones your body can actually make all by itself. But the essential amino acids? Nope. Those have to come from the food you eat. Your body just doesn't have the machinery to manufacture them from scratch. And here's a really cool chemical trick they can do. Because each amino acid has that acidic end and that basic end, when you put it in water, it acts kind of like a tiny magnet. It becomes what's called a zwitteron, a molecule that has both a positive charge and a negative charge at the same time. This dual charge is absolutely crucial for how they behave later on. Okay, so we have our 20-letter alphabet. Now what do we spell with it? We spell proteins. Proteins are the microscopic machines that do all the heavy lifting in your cells. They're enzymes, they're transporters, they provide structure, they do pretty much everything. To build a protein, these amino acid letters are connected one after another by these incredibly strong links called peptide bonds. This forms a long, long chain called a polypeptide, which is the fundamental backbone of every single protein in existence. But here's the thing. A long, floppy chain all by itself is totally useless. The sequence of amino acids is just the raw code, the basic instruction. The real magic, the part that actually unlocks the protein's function, is how that chain meticulously folds itself into a precise, complex, three-dimensional shape. So let's break down this folding process. The first level, the primary structure, is as simple as it gets. It's literally just the order of the amino acids in the chain. Think of it like spelling out a word, one letter at a time.
Okay, next up. That simple chain starts to form localized patterns. Parts of it will begin to twist into these elegant coils called alpha-helices, while other sections might fold up into zigzagging sheets called beta sheets. This creates the first layer of the protein's complex architecture.
And this is the big one. The tertiary structure is the moment the entire polypeptide, with all its little helices and sheets, folds up on itself to form one final, stable, intricate 3D shape. This is the shape that gives the protein its specific job, and it's all dictated by how the different amino acid side chains attract and repel each other. And finally, some proteins are team players. The quaternary structure is when multiple, totally separate polypeptide chains have to come together to form one giant functional machine. A classic example is hemoglobin, the protein that carries oxygen in your blood. It's actually made of 4 distinct chains, all working together in perfect harmony. So we've firmly established that a protein's intricate, folded shape is everything. It's the whole ballgame. So what happens when that shape gets destroyed? What happens when the whole structure just collapses?
Well, that process has a name: denaturation. Things like intense heat or big changes in pH can break all those delicate bonds that hold the protein shape together. The protein literally unravels, losing its complex folding. And when it loses its shape, its function is completely gone.
I mean, just look at the difference. On one side, you have the native or natural protein, perfectly folded, active, ready to do its job. And on the other side, you have the denatured protein, an unfolded, non-functional, inactive tangle. The sequence of letters is still there, sure, but without the correct shape, the message is just gibberish. You have seen this happen 1,000 times. When you crack an egg into a hot pan, the clear, liquidy egg white turns solid and opaque. What you are literally witnessing is the heat irreversibly denaturing the main protein, albumin. You can't uncook an egg, right? And you can't refold that protein. And that brings us right back around to our central rule: structure determines function. But it leaves us with one last really crucial question. Since that final 3D shape depends entirely on the sequence of amino acids, what happens when there's a single typo in that sequence? This is how one tiny mistake at the molecular level can lead to huge consequences for an entire organism. Something to think about.